I am starting recording now, and that should be going. So, as a introduction, we are so pleased today to welcome LaNell Williams from Harvard University. LaNell is a PhD student and an NSF fellow, and she's working in the Manoharan lab. Um, I asked her how to pronounce LaNell's name, but not how to pronounce her advisor's name, so I apologize for that. She studies bulk light scattering methods for the study of bacteriophage and virus self-assembly. She is also one of the volunteer organizers for Black in Physics Week, which is happening this week and still has a lot of amazing events planned through Saturday. And I will be once again posting the link for that in the chat once it's started. She's also the founder of the Women Plus of Color Project. She's a steering committee member of the APS IDEA and an APS counselor for the Forum of Graduate Students and Forum of Early Career Scientists. So please join me again in welcoming LaNell, and let's hear it for virus self-assembly.
Thank you for introducing me, Luna, and thank you guys so much for inviting me here. And I definitely want to thank Black in Physics in particular for allowing me the opportunity to get a chance to talk to you guys a bit about my research in particular. This is a very, special week that's sort of geared towards like celebrating the various like Black physicists that exist in our community. And it's really an honor to be here. So give me 2 seconds so that I can share my screen with you guys. Right. Great. Does everyone see me? See my screen? Okay, great. I was having issues previously when I was presenting and I would start my presentation and everyone would say, wait, we don't see any of your slides. But again, yes, my name is LaNell Williams. I'm currently a graduate student at Harvard University in the physics department. And I work in the Manoharan lab studying virus self-assembly. And today I'm going to talk to you guys a bit about what I do in terms of my research. And so that's understanding the assembly pathway of RNA viruses in particular, and the way that their capsids sort of assemble around their RNA. And the other thing that I want to briefly touch on also during this talk is why physicists should, care about understanding physics from biological systems, but in particular, why viruses are really interesting physical systems to actually study. So it's no surprise that as a result of the COVID-19 pandemic, everyone all of a sudden, cares a lot about virus research and virology in particular as a result of the pandemic. And rightfully so, we know, to date that over 41 million people across the globe have actually been infected by the virus, and over a million people have actually died globally. 200,000 of those are in the US alone. Um, and statistically, we've been also tracking, not necessarily my lab, but the CDC, Johns Hopkins University in particular, have been tracking the statistics for various identities as well, looking at how this pandemic has impacted low-income communities, minority communities, and high-density populations as well. So, it's not a surprise that we care a lot about viruses right now, especially. And as a result, there's been a slew of research, especially in medicine and in biology, to think about ways we can combat the virus, better understand viruses in general, et cetera. And so, it's no surprise that papers are coming out weekly and daily, on this specific subject to not only do good science, but also combat a pandemic in real time. And although medical professionals and biologists in particular have really been at the forefront of making huge moves towards understanding viruses and attempting to combat the pandemic in particular, there have been, some physics labs as well who have, taken the time to think about what cool physics we can learn from this pandemic and how we can sort of contribute to combating the pandemic and learning more about viruses as physicists as well, just to give you an example. And although the pandemic is a perfect example of how some folks in the mathematical sciences and physical sciences have begun to think about ways to contribute, there have been physicists throughout history that have cared about viruses in particular. There are papers that date back to the 1940s and 50s that look at the studies that actually look at the structure of viruses and the way that they assemble. So this isn't to say that although across our country, across our globe, everyone's really interested in viruses now and understanding them. There have been folks who have been outside of the medical and biological sciences who have also cared about viruses as well. So what exactly is a virus in the first place? So viruses, the textbook definition is that they're supramolecular structures or structures that have the ability to infect living cells, like the cells that are in our body. And a very simple virus is composed of a few components. The first being a nucleotide or a genome. And that's just, for those of you guys who may not necessarily be familiar, the stuff that makes up our DNA, right? The stuff that gives us our hair color, our eye color, and tells our cells to do certain functions, right? So the virus also has a genome and has that same stuff that, to tell, its cells, what to do essentially. And then the next very important thing that composes a virus is actually the protein. So the capsid protein that encapsulates it. So the virus has these proteins that sort of surround its RNA and protect it from the environment. So that's what a simple virus is. And in our lab, we're very interested specifically in RNA viruses. And all that means is that the viruses that we want to study are viruses that have RNA, right? So there are viruses that also have DNA. But in our case, we're really, really interested in RNA viruses that are protected or encapsulated by this capsid protein. And more specifically, our lab is very interested in a specific virus called bacteriophage MS2. And MS2 actually infects E. coli. The MS2 virus is made up of 180 co-proteins that are all the same that essentially protect or encapsulate the RNA, essentially. So compared to other viruses, the bacteriophage MS2 is actually, pretty simple, especially when we think about comparing it to something like COVID-19 in particular. So I showed you the very simple version of what a virus looks like, but it's much different than what the SARS-CoV-2 virus looks like in particular. So instead of just having a genome or RNA, and COVID-19 is also an RNA virus, just like the virus that we have. But the difference is now, instead of just having one single protein surrounding the RNA, it has multiple proteins that surround the RNA and that sort of form an envelope. So those proteins include the protein membrane and also an envelope protein. And lastly, it also has these huge spikes on it. So if you guys have seen pictures of it, these large spiky proteins that also surround it. So compared to the MS2 virus, which is what we study, it's much more complex. But, because we're physicists and for the sake of understanding physics, it's really, really important for us to stick with a simple system. So we'll stick with very simple RNA viruses like bacteriophage MS2 to try to understand more physics about viruses. So the reason why we're interested in RNA viruses or interested in viruses in general is because viruses are really interesting for two main reasons. One being that we believe that their infection process is very interesting. And we believe that something called assembly or self-assembly actually is a vital part of how viruses assemble and then go on to infect other hosts. So what essentially happens is we have our bacteria or the cell that it's infecting, and our little virus that's much, much smaller than the cell attaches itself to the cell. Somehow the RNA gets into the cell. And those pieces of RNA end up being replicated. So there's many, many pieces of RNA being replicated in the cell. The next thing that happens is we have some of those pieces of RNA that are being replicated in the cell. Some of those get turned into coproteins, so the proteins that protect the RNA. And then the other, thing that happens is that some of those RNAs begin to fold, right? And eventually, you have a bunch of random pieces of MS2 proteins and RNA just floating around randomly in the cell. And eventually, they just kind of zip up into new infectious virus, almost like magic, right? And eventually, there's enough of them that end up bursting out of the cell, and then they continue to, continue the cycle essentially and go infect other hosts. But what we're really, really interested in is this assembly process. So thinking about how the MS2 virus in particular assembles around its RNA. So this is a nice simulation that was done by the Hagen group at Brandeis. I need to get my mouse over here. Here it is. And this is just a nice visual of what this might look like. So this is a simulation, so it's much simpler than what probably actually happens in real life. But as you can see, like the proteins that are surrounding the RNA are beginning to encapsulate it, and the RNA eventually gets inside of the cell. And so we believe that this idea surrounding self-assembly in particular is a major driver of how the virus goes from a bunch of random proteins and RNA just floating around in the cell to sort of forming these like more complex structures, right? The other reason why we're really interested in viruses uh, as well is, the actual structure of the virus is also very interesting, which is what I kind of alluded to before. So not only, is the assembly interesting, we're thinking about self-assembly. Not only is that interesting, we also want to think about, the structures that these viruses form too. So it's not like these, proteins and RNA are just randomly globbing onto each other is the point that I want to kind of get across. So let's bring back up the picture of MS2. So the MS2 virus is made up of 180, proteins that are basically the same, essentially. And those 180 proteins not only have to attempt to sort of fit together in a sphere, which is already a fairly complex shape to sort of fit into. So if you guys essentially take a bunch of beads and put them in a bottle and you shake it around, they're not just going to assemble into a sphere, right? If you attempt to do it yourself. Um, but these individual proteins also have to form hexamers and pentamers. So these additional shapes that then surround and protect the RNA, right? Kind of like what you would see if you looked at a soccer ball. Another way to sort of describe the sort of structure of the virus comes from one of the papers I sort of alluded to earlier. That's by Casper and Klug, who were like a physicist and a biologist essentially that came together that said, hey, viruses are really interesting. Let's try to better understand their structure, essentially. And they came up with this sort of like idea and found that a lot of viruses have what we call icosahedral symmetry, right? And that just means, so an icosahedron is essentially a sphere that's made up of 20 triangles that surround the RNA, essentially. And so that's the kind of symmetry they noticed a lot of viruses tend to have. That and also cylinders, but we won't be talking about cylinders today. So a lot of viruses tend to have this kind of symmetry, which is really interesting. And they came up with this thing that is called the T number, that to sort of explain the size of the virus, but essentially it tells you how many proteins you can essentially fit inside of each of those proteins that eventually sort of make up, the proteins that fit into each of these individual triangles that protect the RNA, right? And I say all this to say that, the structure of these viruses, so the structures, especially for, in particular, the case for MS2, the structures that these individual proteins have to sort of almost figure out, and sort of navigate to sort of surround and protect the RNA properly are not trivial. They're not simple structures. Right? So I talked to you guys a little bit about, virus self-assembly and the structure of wild-type viruses or viruses that are found in nature, and thinking about self-assembly in terms of what happens in nature. But also, we noticed that it's been found that virus self-assembly also happens outside of the host or happens in vitro. So you can also do this in the lab. So literally, if I take free RNA or random bits of RNA and I add proteins to them, they will assemble into virus, essentially. And if you look at images of viruses, so I'm giving you an example here of the CCMV virus, which is also a spherical virus like the MS2 virus. If you look at the image for CCMV in particular that's been assembled in the lab, it looks very, very similar under these images that are taken these images that have been taken. They look very similar to what the CCMV virus actually looks like in nature as well. So they have the same sort of symmetry, they have the same sort of curvature, if you think about it. And this is really interesting because this sort of moves away from this idea that for this particular virus that, there's some sort of like, sort of by design way of like these proteins just kind of coming together as needed. There's some sort of physics that's also really interesting because we essentially have a bunch of random parts that are kind of floating around, this polymer that eventually have to figure out how to make this sphere. And so because this also happens in vitro or outside of the cell, and we can do this in the lab as well, we can say that we can sort of propose that like, well, maybe the virus, like many other like random systems and systems that are under Brownian motion in particular, or just random systems for those of you guys who may not understand, may not know, But these random sort of bits and pieces of proteins and RNA that are floating around, the way they sort of find their way around to protect the RNA may be driven by this idea that we call minimizing the free energy of the system, or minimizing the amount of energy that you use in the system, so that you end up getting a full capsid. And so let me further explain what that actually means. So here I've driven like a cartoon of what we would sort of say is a free energy landscape, which is essentially just the number of states that the virus can kind of explore, like, the different configurations the virus can explore. And so we can imagine that as the virus sort of explores this landscape or explores this space, sometimes it could be just a bunch of random bits and pieces of RNA. If you can see my mouse, hopefully. Sometimes it might begin to form a partial capsid. So it may start assembling and then it stops or something for some reason. Sometimes it might, form a malformed capsid, which means that instead of forming just a sphere, it forms some weird shape, that may not necessarily be functional for it. But we know that eventually the virus or the proteins in the RNA eventually find a state or find some configuration that sort of represents what the properly formed capsid looks like, or represents what a full capsid looks like, right? But this sort of minimization of free energy argument doesn't necessarily, so it tells us, you know, uh, it explains a reason or why this might happen, but it doesn't necessarily tell us how the assembly is actually happening as well. Um, so I think what's really interesting, though, in the picture that I'm trying to show you guys here is that I'm really interested in the fact that there are some states in which we do get full capsids, but there are other states in which we have capsids that are either partial, might have capsids that are partial, or might even form other structures. Um, and so the gist of my project in particular is, trying to better understand not only how the virus ends up forming these full capsids that end up protecting the RNA and then going off, and being some other, form or something like that and continuing the process of infection, but also thinking about, what might cause it to not necessarily do that? What might cause it to form some kind of monster capsid like I have here on the right, or not finish its assembly at all and just stop. And so the first step to sort of answering that question is thinking about, what conditions can I sort of like change or put on the sort of assembly process, thinking about the conditions that sort of promote this correct assembly, so the virus being a full capsid, versus conditions that might promote the other cases where you have a monster or you have something that doesn't finish itself, right? And I want to do this by creating what we would call an experimental phase diagram, which is what I will get into next. So the way that I do my experiments is similar to what I was saying earlier, is that I take free RNA, MS2 RNA, I add proteins to the mixture, and then they essentially assemble into virus, right? Some of that virus might look like the full capsid stuff, and some of it might look like the malformed capsid, or the, partially formed capsid. And, that can change depending on how I sort of, change up the conditions that I do my assembly under. And I'll get into that in a minute. But what we want to do is do this kind of experiment and then measure what we get at the end, right? And so this is sort of my pseudo, or, sort of cartoon version of what my sort of phase diagram would be. And this just describes like, what are the different states or configurations the virus can be in, essentially. And so I believe that, there are some conditions that end up promoting, you know, full capsids that are nicely formed and they protect the RNA properly. Some conditions that promote partial capsids, where most of the sort of stuff that's in our sample ends up being just unfinished viruses that have not finished their assembly. And then other conditions that might promote these monsters that may happen. And figuring out those, conditions and what we should change is something that we have to think about. So ideally, what we do for our experiments is changing sort of the variables that are in our buffer condition. And so the buffer, so the sort of liquid that we essentially place the viruses in when we're doing our experiments, are basically set and constant, and sort of we start off with an ideal environment for the virus, essentially. And then what we go in and do next is just tweak the things that we can change. And three things that we can realistically change in our buffer conditions, essentially, are the ionic strength, with is just we just add salt to the sample or take away salt from our sample, right? And that somehow affects the assembly. We can add more proteins to the sample, or we can add more RNA to the sample. We can adjust any of these variables and see if we can sort of come out with this sort of phase diagram that represents all the various states that the virus might, possibly be in. So once we do these experiments and we change the conditions, we need a way to measure it. And so we're going to take advantage of two techniques. One of them being gel electrophoresis, which is very commonly used in biology, and which will give us some visual representation of what might be in our sample. And the other being dynamic light scattering, which gives us some quantitative data about how much stuff is bigger or might represent malformed capsids, how much stuff might be smaller or represent stuff that might just be partial capsids or individual proteins, and how much stuff might be actual MS2 virus, right? And we just want to know the size of stuff, and that's what DLS gives us, or dynamic light scattering. So I'm going to first start with our gel electrophoresis, and that gives us qualitative data, so visual data that we can use to verify the quantitative data that I will show you guys next. So I want us to imagine that I've done 4 experiments, right? 4 experiments where I take protein, RNA, mix it together, and I get something, right? And in those 4 experiments, I end up getting, these 4 samples. So sample 1, sample 2, sample 3, and sample 4. So the way that gels work essentially is we have this thing in a gel called wells where you place each of your samples in, right? So sample 1 will go in the first well, sample 2 will go in the second, and sample 3 and 4 will go into the third and the fourth well. And then we place those samples under an electric field. And all that does is cause the sample to kind of travel down the gel, right? So as the samples are traveling down the gel, the bigger stuff moves slower and the smaller stuff moves very, very fast. So that's how we can get an idea of what the size is in our gel. So if we look at samples 1 and 2, right, we have MS2 virus, right? And so we notice that our bands, so the lighter sort of gray represents the sample. So the sample is traveled down, right? And to a position that represents a size that represents the MS2 virus, which is kind of in the middle, right, of something really big and something really small. The next sample we're going to look at is the third sample from my third experiment that I've taken. And that sample essentially represents stuff that's really tiny, like tiny bits of RNA, the small stuff that just flows right through it to the bottom. And then the last thing that we look at is sample 4, which has all 3 essentially. And so sample 4 looks like it has some big stuff in there. If you look at it, look at the actual sample here that I've kind of drawn, some stuff in the middle and some really small stuff too. So you see each of those lines represented in the gel. And so to give you an example of what some of this looks like in my real data, so on the left of this line, I run some experiments that essentially tell me, hey, this band or this area represents where the MS2 virus lies. And then I do a bunch of experiments where I increase the RNA concentration. So we're going to take the first variable, one of the variables that I said I wanted to change, and I want to change RNA concentration. And so our RNA concentration increases in this direction, right? And we find when we look at our bands, voila, we have some MS2 capsid being formed in each of these experiments. And then the next sort of set of bands that are above that represent malformed capsids or things. We don't know if they're malformed capsids or not, but stuff that might be larger, than the MS2 virus that could be malformed capsids in particular, right? And then we do the exact same thing, but for increasing the protein concentration. And we see something very similar there. So that is gel electrophoresis. That's the first technique. The next technique we're going to take advantage of is dynamic light scattering, which will give us an idea quantitatively of how much big stuff is in our sample and how much small stuff is in our sample. So DLS, or dynamic light scattering, starts off by taking the sample. So we've done our 4 experiments, we've done our experiments. And then what we do is we shine a laser at it. We shine light on it. Some of that light is going to be transmitted or passed right through the sample. Some of that light is going to be scattered off. And so when we take this measurement, what we're measuring is the light that's being scattered off of the sample. but more specifically, we're measuring the fluctuation in that light as particles are diffusing around or bumping into each other in the sample, right? So for instance, if we took a sample and we have a bunch of small stuff in it and we wanted to sort of get an idea of what the intensity versus time is, or the fluctuations of our signal as small particles are moving around, the sort of curve would look something like this, because these are very tiny particles that are just bumping around, into each other really, really quickly, right? But if we look at a sample that's in the middle, like for instance, for the MS2 virus, you notice that that signal ends up getting more broad. And that's because particles are diffusing more slowly around. So bigger stuff ends up kind of bumping into each other a bit more slowly, right? And then for really big stuff like our monsters that we want to, I'll call them our monsters for now, the signal gets even more sparse because now things are moving even slower in the sample. And that's what we end up measuring. And so what we end up doing is we take these curves that represent our small, large, and large particles and our MS2 capsid or things in the middle, right? And we invert that data. I just want to briefly mention that's also something that I'm also working on, because the inversion of this sort of these curves for samples that have more than one thing in it, so samples that have big things and small things in it, is really complicated, and is something I'm also working on. But we invert that essentially, and we end up getting at the end uh, these graphs that represent the intensity of our signal with respect to the size of the particle. So for small particles, we end up getting a curve that's lower on the end of our scale. For larger particles, we end up getting something kind of in the middle, right? That represents something that is the size of the MS2 capsid, just to give you an idea. And then for big particles, we end up getting getting a signal that's further to the right, essentially. And then I guess what this looks like if we just take the sample. So this is a sample of what my data looks like when I take it. You essentially see here that I have a peak here that represents the MS2 virus, which is about 30 nanometers. But I also have a peak here that represents something else. It might be a more malformed capsid, could be dust. But this is what we're attempting to do with this DLS data. And it's pretty tough to do with when you have more than one size particle in your sample. So if we want to combine this data to kind of begin to build our phase diagram, we'll do something like this. So let's take one of our columns, for example, that has one band, right? And we compare it to our MS2 virus here, where we see a peak that corresponds to the size of the virus, essentially, which is about which is about 30 nanometers, essentially, right? So this is, a sort of visual representation of what what the size of the MS2 capsid is, and this is what it looks like quantitatively, essentially, right? And we can use this data to begin to build our phase diagram. If we look at a different column, like the one at the end, right? If we noticed in our column here, we have two bands here, one for the MS2, right? And then one that represents something that is a lot bigger than the MS2, something that might be a monster. And if we correspond that, so it's essentially what we're trying to do is correspond that to what we see in the DLS data, which gives us a quantitative representation of that. And we see a peak here that represents the MS2 virus, right? That's about 30 nanometers or so, which is within the range that we want. But then for that other band that we see here, we also see something else, right? That's much larger. And that's also represented in the gel. And we do the same thing for both the increasing RNA and protein concentration. And what I begin to build is this start of a phase diagram that essentially places the percent yield of the assembly products that we get in the end. And what I want you guys to kind of take away from this is that what I see in this graph here, so when I plot my capsid protein concentration versus the RNA concentration, I do see a region in which I see 100% or close to 100% full MS2 capsids. And then I also see a region in which I see about actually 50-50 or so capsid to the malformed capsids. But I want to emphasize that this is just the start of what of more experiments that I've been doing and will be continuing to do to continue to feel out this experimental phase diagram, right? And I want to bring it back to what I showed you guys earlier about what we're attempting to do. And essentially, what we want to do is, again, these are the methods that we're using to essentially to put together to begin to build this diagram. And what we essentially want to do is build a more robust version of this thing that we have here on the left, that will hopefully represent, as I said earlier, regions that that promote full capsid assembly, regions that promote malformed capsid assembly, and regions that promote the smaller things or incomplete assembly. So in conclusion, I have seen from some preliminary data and from some data I've taken before that as we increase the RNA and capsid concentration, we do begin to see some of those regions. And, the sort of idea that there are metastable states or other states that the MS2 virus can be in. And I want to emphasize that this is really important because if we try to better understand the conditions that promote full capsid assembly and promote these other structures, we can also think about disassembly as well, or thinking about the conditions that pull viruses apart. And that could be extremely important for research in virology and even like methods for developing vaccines as well, if we want to think more broadly. In terms of immediate next steps, again, creating a more robust phase diagram. I'm also working on some DLS experiments, working on an inversion model for the dynamic light scattering data that we get from our DLS instrument. And then I also had done some experiments as well that give us some kinetic data if people are interested as well, thinking about like what essentially like changes the assembly rate of the virus assembly when we adjust our buffer conditions. And so I want to thank Yale for having me today. I also want to thank Black in Physics for allowing me to also speak about my research today. My funding sources, Harvard University, both the Physics and Cs department, my lab, and everyone that decided to come out and support me today. And I guess we can open it up for questions.
All right. Thank you, LaNell, for an excellent talk. I'm an astronomer and I feel like I can follow virus self-assembly, which is excellent. So anybody have questions right now? VJ?
Hey, LaNell. Thank you for your talk. I really liked it. It was very clear. And I appreciate the motivation and sort of the contextualizing in the context of physicists and how we can sort of contribute to a problem that we probably would assume is outside of our scope. I'm interested to learn a little bit more about the dynamic light scattering experiments that you talked about. And just wanted to understand in the graphs that you showed of, I guess, particle size versus intensity. What assumptions go into calibrating the size of these particles, and sort of what type of scattering framework you use to extrapolate the size of the particles?
Yeah, I'm glad you asked that. I have backup slides on this because I knew that people when I show you this, you're gonna realize how much I took out in terms of understanding like the dynamic light scatter. So these are my kinetic experiments, but this is like the sort of full DLS sort of spiel here.